The overall goal of this experiment is to synthesize and characterize keratin-based nanofiber for biomedical application.
This method can help answer key questions in the biomedical engineering field, such as wound healing, drug delivery, and engineered tissue such as liver, bone, and muscle. The main advantage of this technique is that it is cheap, efficient, and can be upgraded to large-scale production.
The implication of this technique extend toward treatment of wounds and delivery of drug for tissue regeneration. Though this method can provide insight into the advantage of using Electrospun nanofibers, it can also be applied to other systems, such as tissue regeneration. Generally, individuals new to this method will struggle because it is difficult to make a polymer solution that can generate nanofibers.
Procure approximately 20 grams of human hair clippings that have not been chemically treated or altered. Hair can be any length. Wash the hair thoroughly by hand with warm water and soap. Use deionized water for the final rinse. Put the hair into two 600 milliliter beakers and place in an 80°C oven for 1 hour. Once dry, divide dried hair evenly between the 600 milliliter beakers, placing 10 grams of hair into each beaker. Do not allow the hair to fill more than 500 milliliters. Next, pour 500 milliliters of peracetic acid solution, or PAS, into each beaker